Hello there, how are ya? Welcome, I'm Bill. Welcome to session 2 of the AP Bio. I will be your host for less than an hour of a lot of material that we're going to talk about. Just remember that here at the Princeton Review, we're here for you and we're here to help you achieve your goals. So let's get rolling, okay? If you saw any of the videos, the video last week, then you'll see that there's a little bit more I was talking about in terms of myself at Princeton Review. I've been here about 7 years now, and I teach a whole variety of things. And this is how you can contact me. All right, let's talk about a little bit more in detail about the AP Bio exam. There's going to be a lot of material, and it's going to be cursory, but it's for a reason, because of the amount of material that you need. Now, I'm going to give you the link to the previous video in a second, but this is just information that's also provided in the first video as far as when the test is. It'll be 2 questions. I think by now you probably know that, 25 minutes and 15 minutes to go with. What we're going to talk about today as far as the material, and like I said, there's a lot of material packed in today, is we're going to start off cursory with a little bit of the cell structure and function, and then get into cell communication, and then talk about energetics. There's also a section that I'm going to talk about in terms of the statistics that are relatively important to know that you can use. Again, it's all just a cursory. You'll have to look up in all your references and so forth. And there's a ton of information here, as you could see in the College Board, and in these other prompts here. And again, this is taken out of the last video as well in terms of the AP material. So you can take a look at that. Okay, same story. It's going to be 2 free response questions. And here's the link to the first video where I do a little bit more of an introduction into the strategies. I think we got into a little bit more of the chemistry of life. Okay, and that's what we talked about. Now, these percentages here, remember the key thing here is, if you didn't know, unit 7 and 8 aren't going to be on there. There's going to be the ecology, the population, and so forth, and climate kind of information. And we covered all of this in the first video in terms of that. What we'll talk a little bit about today, and these percentages are probably raised, especially since they dropped to 2 units. So you could say more than 13%, if you will, would be on structure and so forth. Then we'll get into cell communication over here, which is probably, I don't know, at least 15, maybe more than that as far as a percentage. Like I said, because that's what they would do. And then cell energetics. Now, the thing is that you have to be careful here is there's only 2 questions. So it's more of a probability of what you would have. But it's very difficult to say, okay, you're going to get one of these and one of these in terms of that. So the point is just trying to figure out the kind of stuff that you need a little bit more refresher. And that's what I'm here to do today, to give you a little bit more of a refresher on things so that you kind of gauge where you're at. You're not going to know everything all at once. You're just going to have to figure out where you're at in terms of how much of the stuff that I remember. And I know that further stuff isn't going to be on there due to our current COVID situation. All right, let's talk about structures. Again, structure and function. The bottom line is everything is about cells. And there are many reasons why there's compartmentalization, as you know. And the second reason we can't be giant cells is because there's a big surface to volume ratio. And there's many things in the body that has to deal with surface to volume ratio, et cetera. So therefore, that's the reason why the cells are the way they are. 2 types, prokaryotes and eukaryotes, okay? Of which, you know, bacteria, the prokaryotes that look like this. And make sure that you're familiar with pretty much the basic terms that are associated with prokaryotes. Eukaryotes as well, okay? And each one of these we're going to simply touch on, okay? Just because you need to just be able to refer to it depending on what the question's going to ask. Plasma membranes are very, very important, as you know. There's a phospholipid bilayer that's important. Make sure you understand the hydrophobic, hydrophilic, philic meaning like water, and how the phosphate heads face in and out of that membrane. Noting also that a knowledge of the idea of the semi-permeability is also an important topic in terms of that. Anything that's going to be larger or something that's going to be hydrophilic is going to have to pass through some other kind of tunnels, which we will just mention again. So that's the kind of thing, understanding the lipid bilayer of importance. Fluid mosaic model, I'm sure you've heard about it. You have your proteins on the outside, you've got some that are embedded, basically floating in, and you have carbohydrate side chains that are out. So you have the outside and the inner, and a solid understanding of that would be very, very useful. You have transport proteins, some pumps, and I'll show you an example of one that was on a test last year that I wrote up here. So that's going to use an active transport. Some other ones are channels. That's going to let basically selective is the key thing. And then some are glycoproteins or cell surface markers that are going to play a role in things like adhesion, especially when you're looking at things like blood and so forth. Okay, the nucleus is our big player here, as you know. We got these larger structures called chromosomes. We'll get into mitosis and meiosis and follow the hereditary and all those kind of things next in the last lecture. I saved that for the last. And your RNA is made in your nucleus, nucleolus. Okay, you should associate ribosomes with protein synthesis. You have your ER. Notice the rough is the studded, as you know, with the ribosomes, and the ones that don't are the smooth. Golgi bodies are also important for packaging, distribution of materials. Mitochondria, you probably heard this 100 times, powerhouses. They're going to convert your energy and those organic molecules that we talked about last time. And obviously the big one is ATP. Okay. It's easy to understand because it kind of looks like basically an oblong shape with a zigzag inside it. It's obviously a lot more complex than that. There are many, many ways in bio by which you have this folding in terms of that. The villi, the alveoli in lungs, which of course is a big player right now with the COVID, and root hairs in plants. The idea that there's a lot of folding that's going to increase the surface area. So when you fold something, you give the cells a greater chance to be able to look around and attach and adhere and interact and so forth. Lysozymes have to do with digestive, right? All kinds of things. They're eaters of the old, as we say, and centrioles are important in cellular division, okay? And note the microtubules, which pull the replicated chromosomes apart. Okay, well, then we round off our discussion with vacuoles, and I'm going to give you an example of a vacuole in a flower kind of thing as an example that was an exam last year. And then perioxysomes and your cytoskeleton also. Again, these are just, it's more knowing the language and being comfortable enough that you know it, that there won't be any issues in terms of that. When it comes to cytoskeleton and cilia and flagella, then, like I said, we broke it into micro tubules and microfilaments. And basically you have, in terms of the microtubules, this is a nice little chart set up and what's assisted, whether it's muscle contraction, as you know, or any other kind of movement. Cilia and flagella are also the thread-like structures that are in the single-celled organisms. So separating that would be very helpful. I think we get the idea that the cell wall is is the distinctive factor between the plant versus animals. It's going to give you support with the plants, the protists, the fungi, as well as the bacteria. And of course, the other one is usually a vacuole is going to be your distinctive fashion. So you get those two together and you're in great shape in terms of understanding that. The structural characteristics of different cell types, if you just summarize, are nicely summarized here. If you get the gist of this, and you won't be asked to reproduce anything like this, but this, if you separate this and you're able Well, one of the things that I pointed out to last video is one of these that you need to do now that you have time to study is you have to basically, and this is what the way I learned everything, is to take a blank sheet of paper and then write down what it is that you need to know. If you're able to do it on a blank sheet of paper, that means you understand it. So you can make this chart up and just go through all the different structures and see what's what. Now, I'm not saying that you'll have to reproduce it for this particular exam, but my point is it gives you a good handle of your understanding of it, because remember, it is stuff that you're going to have to write in terms of an explanation, and if you understand it well enough, then it's super easy to write. And again, we'll touch on a couple of the strategies to do that in terms of that. All right, continuing on, we were talking still about cell structure. Now let's talk about a little bit more of the function, which is transporting, okay? The way things occur across a cell membrane depends on a couple of things: the semi-permeability and, of course, the size and charge of the particles. If you're dealing with something that's hydrophilic, that loves the water, then the bilayer won't let it pass without any kind of assistance, so that's going to be facilitated. Okay, facilitated means helping, okay, in terms of that. And that depends on a whole bunch of proteins that act on there, and aquaporins is also water-specific channels. And that's a good enough point. Simple diffusion is also something that, although it is simple to understand in the idea of diffusion, it's also something that you want to make sure that you know the difference between that, okay? If you're dealing with something that's hydrophobic, I can't tell you the chances that you'll see hydrophobic versus hydrophilic, but as long as you're armed in both regards, you're all set. And basically, because the small nonpolar is just going to drift right through the membrane without any issues, that's called simple, okay? And passive, as you know, is something where it's not going to be anything. It's opposite, basically, of active. It's not going to need any outside energy as you move through. Okay, and this is a simple experiment. There is a lot of mathematics to diffusion of which you do not need to know. All you need to know is just the qualitative aspects of this, nothing quantitative. And anything that would be straightforward, because it's not necessary in this particular arena. Of course, you know of osmosis. You've heard that. That's when you're dealing with water. So when you deal with osmosis, you're thinking of water as it moves down a concentration gradient, which just basically means it wants to go basically from where there is less to where there is more, okay? Because if something is less solute, it's going to be more aqueous, right? More watery, I guess is the right way to say it. And if something has got more solute, right, that's going to be more of a concentrated, right, in terms of the solute. Or you can look at it in terms of the water as well. It's going to have less water because it's got more solute in it, okay? And that's how you're basically dealing with that. So, and it says here, it says if water is moving to the dilute any of the concentrated solute particles. And there's many applications to this in the real world that you've heard of. Get our slides. Okay, tonicity. The key thing with tonicity, and make sure that you understand it, is being able to use the word. Okay, and again, you'll see in a second a good example of that. If something's iso, that means you've got some equal. If something's hyper, then it's got more total dissolved solutes than the cell, and hypo is going to be less. Water potential is also another thing that you can use. I'm sure you've seen this before, but explaining basically water potential as the potential energy in the water. All right, so the eagerness, if you will, the desire of water to flow from high water to an area of low water, and that's called water potential. Like anything you may have seen in physics in terms of a potential to do that. So when you add a solute, you're going to lower the water potential. That means that water is less likely to leave the solution and more likely to flow into it. Okay, now you need to keep that in mind in a few minutes. Okay, now we've got active transport. To really go into in depth of it is definitely not purpose here, but basically anything that's going to go against call natural flow. It's going to have a lot of resistance towards that. Okay, so in order to be able to do this, you need energy, and that's where the ATP is going to come in. Okay, so you know your sodium potassium pump that's going to take out or usher out is a really good way of describing it. 3 sodiums and bring in 2 potassiums. Okay, and this here also comes into play, resting membrane potential. Something to keep inside or keep in mind in terms of a potential question that you may get. You have endocytosis and exocytosis. Sometimes you take the large particles, they come out of the cell, that's exo. And if you form a pocket, it's basically endo. You think of the pinching in and so forth. That's all passive, okay? So this is a quick summary. We talked about all the different parts of a cell. Again, it's cursory, but you do need to make sure that you're able to explain each one of these in the event that you need or you're pointed to in a question. Each one of these and its basic function is important, okay? And this is a long list, but pretty much of stuff that we just talked about, but also the same kind of thing that they would expect. In terms of function and so forth, again, note the layer, the bilayer, note size and polarity is very important. And part of your free response is, since you are writing this, is to make sure that you explain things. So you'll always note size, polarity in some kind of transport of any kind of membrane. And any of this transport that we talked about, again, you just look at it as which has got a concentration change, which direction would it go, what about the dependent in size, and so forth. Okay, now, what I've done here, I know it looks kind of scattered, but it's the best I could do because I'm not allowed to reproduce something that the College Board had. But basically, if you just look, and what I'm asking you to do is just look at this problem, okay? This is a problem that basically says, changes in petal cells during the flower opening, okay? And you have 2 things. Here's a bud and here's an open flower. So you got 2 things. And basically the problem is there's a specific type of flower, okay? It changes from red, as you could see here, to blue, okay? Depending the bud or the open flower. What happens is it changes from red to blue and the petal cells tend to open up during the flower opening. And this thing is blue because of anthocyanin, which is found in the petal cells, okay? Now, petal color is basically determined by the pH. Now, the numbers here, let's just say that this is distinct enough. The pH of the bud would be 6 when it's a bud, of the vacuole, of course, and the vacuole pH is 7 when it is an open flower, okay? So this is a model basically of what happens in terms of the channels and transport proteins and the proton pump. As you could see, I tried to draw this distinctive for you so that you could see. So you've got the transport protein, which in this case, in the bud case, is inactive. The transport protein in this case is active, so it's shuttling in the potassium. Out comes the hydrogen, okay? And yet you also have a potassium channel bringing in the potassium, okay? The proton pump bringing in some of this, and again, as such. Now note that this transport, again, is inactive, but it is working here. And as you could see, this draws in water. Okay, so having said that, and I get this, you know, I have to switch slides here, but I understand the idea is, the keys to doing any type of question, whether it's going to be, this is a smaller question, whether it's going to be the longer question or the smaller, depending whatever the case might be, is to make sure that number one, you answer the question. You have got to do that in any way, shape, or form. Now, most of the time you're going to think quantitatively. In this case, there's a little bit less, but the idea is that you need to say this number, this would be greater, this is less, and that's the idea, to think in quantitative means. To be specific, what trumps everything here is clarity, being able to express, but of course you also have to be accurate. You get that from all the knowledge that you're learning. Vocabulary usage, the more vocabulary, in this case, all the vocabulary we just went over, better you are. And then finally, the big ideas, of which are just the general ideas that they're trying to bring up. You can find these in the guides. This is also something to think about, but it's just, again, in the back of your mind. Now, what I've done is I've left this blank so that I could just basically tell you, here's a couple of things. Basically, the question's asking, identify. Now, I get it that you don't have it in front of you, okay? But the question is asking you to identify the one component in this cellular model, okay, that's going to be responsible for the increase in the pH. Now, I wrote this in capitals for a reason. These verbs are very, very important to understand, because that's the question that you have to understand and you have to address. So it says, which is the component that's going to be responsible for the increase in the pH of the vacuole? Okay. During the flower opening, which we just mentioned, and describe. Okay. So, and in that term, if you go to the first video, you'll see these verbs we listed out, but you can also get that information from the College Board. The component's role in changing. So this component in changing the pH. So it's not just what's involved in the increase in the pH, but also the changing of it. Okay, that's one part of this. I just want you to focus on what the question's asking. Okay. Will constructing models, graphs, and tables, the question is, will constructing models, graphs, and tables still be on the new exam? If it's going to be anything on the longer side, it would be the first question that's going to be worth a little bit more, but it would definitely probably be abbreviated because there's a lot. Now, as far as constructing the graphs and the tables, no, because this is just a written response. And that's my understanding in terms of written response. But describing a model like we have here would most likely be what you would have to do in terms of that. Okay, it's not requiring you to actually draw anything anymore. But if you were going to plot, you would describe it, my x-axis, y-axis, and so forth. Okay, good question. Now, the second part of this, so that you get, is basically saying that a researcher is claiming, all right? So I'm writing this like this so that you understand. A researcher is claiming that activating, okay, activating the potassium hydrogen transport protein causes the vacuole to swell. Let's just say causes, we're assuming that the vacuole is what we're talking about, causes swelling with water. Okay, and what you have to do now is provide reasoning. So this is how they write it. Now, this is a short question, don't get me wrong. It's not a too long question. But as long as you understand what they're asking you in terms of this, which is, first of all, identify the thing that's dealing with the component of this model that's responsible for the pH increase, and then describe that component and how it changes the pH. And then the second part of this question, you know, and it could be labeled Roman numeral A, Roman numeral B, etc. You can go this way. All right. So in terms of that. Now, getting back, I'll give you a second to look at this. Hang on one second. Okay. So looking at this, basically, what you have to write and literally write would be the first part, when it's asking you to identify the cellular component in the model that's going to be causing the increase. Anybody can tell me what that is? So the question is asking you basically identify the cellular component. Which one of these? Proton pump, your potassium channels, you've got all this. Which one of these is going to be responsible for the increase in the pH? Any takers? And then basically you want to explain it. Anyone? Okay. There's probably a delay, so I understand that's not a problem. All right. Basically, what you want to say in words is basically that the potassium hydrogen transport protein is responsible. Now, there's many ways that you could say and describe how that's going to happen. And we could do this over here. There you go. So it would be the, and I'll change colors here so they could help you out. Okay, basically it'd be the potassium hydrogen. Now, you just don't write that. You would have to write it out a little bit more in detail. Okay. And why would it do that? Because it's moving this out of the vacuole. Again, you would be writing this out in sentence form. No, actually, yes. Actually, it would be the, yeah, it would be the transport protein because it's moving the hydrogen, because you're talking about the hydrogen out, and that's going to change your pH. Okay. Because basically you're changing the pH by removing the hydrogen ions, okay, that are being brought in by the proton pump. So you make a good point. The proton pump is bringing in the hydrogen ions. So I hope maybe I read that. No, that's, yeah. So, but what they're asking you is what's playing a role. That would be the transport protein here. So this is a perfect example of how you need to fully explain out the thing, that the transport protein is, the pH of the vacuole is increased by the transport protein. Okay, and that changes pH by removing the hydrogen ions out, or I should say removing the hydrogen ions that are brought in by the proton pump. That means and this is very important, you have to talk about how this decreases, which then increases the pH. You see, this is the kind of understanding, but in a step-by-step manner that you need to write in a clear sentence. And then you could just basically talk about a couple of other things. But again, you don't need to cushion it too much. Just basically say that the potassium and the hydrogen are going in those directions. Now, the second part of this is telling you, here they give you a claim. They're telling you somebody's saying activating this protein is going to cause this thing to swell. Okay, so now it's provide the reasoning behind there. And this is something along the lines that you could say, you could talk about movement going in. The vacuole is also accompanied, as in osmosis, which means water. Okay. Because moving potassium in, and again, I'm not writing it out, it's faster to explain, but moving potassium in, if you notice, is going to, into the vacuole by the pump, is going to decrease. And here's another one that you can use, the idea of water potential. Now, you can say water potential goes down. You could say that the vacuole is hypertonic, right? You could say that vacuole is hyperosmotic. Okay, you could say a couple of other things in terms of that. All right, no matter which way you want to look at it, you could say that, but you can you could say decreasing the water potential, and that means the water is going to move in with the potassium. So you got to say that water is going to move in with the potassium, okay, to balance out any of the changes in the psi, excuse me. You could do this, but I would rather just write out the word water potential. It's probably stronger, so that you're going to reach equilibrium. You should always have a discussion of equilibrium when it's pertinent to what you're dealing with, okay? And while that's happening, that means water's going in and the thing is swelling with water, okay? So that's how you would attack this kind of thing in terms of a written response. And hopefully you have a better idea of how to do this. They'll give you allocate the appropriate points for saying the protein, etc. Pretty much what I've written in blue. But again, it has to be written, in something that's a coherent discussion. Okay. So hopefully you found that helpful. That's a good example, and it's a pertinent one that was actually on the actual test. Okay. Now let's move to energetics, and the reason why I want to move to energetics, because we talked enough about transport, and I just mentioned a couple of things about it. The key thing here is, let's see where I'm at here. Hang on one second. I just got make sure my. Okay, apologize for that. There we go. All right, so talking about cellular energetics is the next phase of importance here that we're talking about. Okay. Now, mentioning a couple of things, the same kind of thing. When you're dealing with bioenergetics and chemical reactions and so forth, it all has to do with energy, change in energy, and the standard energy can never be created nor destroyed. So that's your golden rules. It's not going to be the only thing that you say, but it's basically the idea of what you got. You have exergonic, is how they say it in biology, endergonic reactions. It all has to do with basically know both directions that you're going in terms of reaching. In chemistry, you know this a lot more with activation energy, endothermic, exothermic, and so forth. You still need to know the basics behind there. Here's your activation energy, which is that portion that you basically have to basically get past, overcome, etc., before you go down. All right. Once you reach this activation energy, everything is downhill, and you reach a reaction state. I should say a transition state, which is the tough part. And there's a lot of things that go on there in the transition state, which won't be tested. You need to know an enzyme, and you need to know the basics of it. It's going to speed up without getting involved in the reaction. It's got specificity, and substrates, all right, are the targeted molecules, and that's what they call them. Okay. They've got different jobs, if you will, in terms of the active site. You form the active substrate, and an understanding of this in the basic form is pretty much enough that you need in terms of that there isn't anything that I've seen that's going to ask you, some real heavier mathematical type of things. They don't do that on this test. Comparisons like this are perfect, okay, and make fantastic explanations for when you're writing something that has to do with enzymes, which is basically forming these temporary, increasing the rate of reaction, complexes, and stay out of the reaction, don't get consumed, and so forth. But they do not change the reaction or make anything, outlandish in terms of that. Cofactors and coenzymes are also important to note. Okay. You definitely have to deal with temperature and pH. Those are the first two things that you would describe in any kind of enzymatic reaction when they talk about things. If something's denatured, a denatured protein, that means it's damaged. Q10 is also in your AP biology equations format thing here as well. Also. So it's not difficult in terms of understanding it. And obviously enzymes work at a specific pH. You have allosteric sites, okay? Sometimes when they're binding other things, you call that allosteric. The idea of competitive inhibition and the idea of basically you're going to fit this active site on the enzyme and compete, therefore blocking the substrate from getting into the active site. That's your competition. And also note that you have non-competitive inhibition if you have an inhibitor, and you should know the idea of inhibitor binding, distorting the shape so that it cannot function, okay? And note that the enzyme isn't going to be part of the catalyze the reaction, which is exactly what's going to happen if you're trying to do that. ATP is a lot of energy, and obviously is very important in cells. This here is your standard equation where you're going to get your energy out of there. It's straightforward. ATP. The key thing here is noting the big differences, especially the sources of ATP in terms of what's going to happen. Okay. Where does it all come from? Well, you already know this, but here's to review: cellular of understanding the absolute details. Here's your overview. As you know, you have light and dark reactions. Okay. Understanding the differences is very important. Here's just your general picture, which you can easily find in other literature and so forth. But understanding the light versus ones that do not, right? The dark, if you will. Okay. The ones that are not going to be light independent, if you will, is very important. And also an understanding of basically within the cells. Okay. There are a lot more things involved in chloroplast structure. You can find that depending on your resources to review. If you look at the absorption spectra in terms of this is just basically a nice summary of that, how well a certain pigment is going to absorb EMR. Opposite of that is going to be the emission. That's going to talk about what is emitted from that. Okay. And things to note, like the light reactions are going to occur in the thylakoid membranes are the important things to talk about. And I get it, this is a cursory view. This is more of a discussion basically on photosystem 1 and photosystem 2 and the light reactions. Okay, so you need to look at each one of these in terms of just the key points. Okay, you have light independent, which is your Calvin cycle. Now they call it the Calvin-Benson cycle. And the important things of the dark, right, the light independent, is that you're going to have this occur in the stroma, right, not the thylakoid. ATP and NADPH are going to be the necessary to fix the carbon, and that CO2 is going to be fixed to form the glucose. So those are the points that are going to be very useful to know. And here's a quick summary of everything where you locate with the thylakoid membrane versus the stroma and what the input is. And this is the kind of information that you can summarize and able to put together. You're in good shape in terms of that. Aerobic versus anaerobic, I think you have a good feel for, or you should have a good feel for that. The aerobic is the more complex because of its 4 steps. The easy thing to do is take each step individually. Make sure that you understand the basics that there are, that, you know, NAD and FAD are going to be your electron carriers. FAD is going to be reduced by 2 hydrogens. Note, FAD is fully oxidized. And that if you have an FADH2, you're going to have the 2 electrons and 2 protons. So the basics of that I'm sure you've learned before in this order, understanding each one, and then it all comes together. Okay, so here's your glycolysis. And like everything else, you got to put in money, right, to make money. So you're going to put in 2 and you're going to get 4 and you're going to gain 2 NADHs. And that's the gist of it. So these are the tidbits that it's in the cytoplasm. You net 2 pyruvics and 2 NADHs. Okay. The second stage is the formation of the acetyl-CoA. And this is in the mitochondria, right, where pyruvate is going to turn the acetyl-CoA and 1 NADH is going to be made. Okay, and there's a chemical formula for that that's written, but you don't, as long as you know the gist of that. Then your Krebs cycle, TCA, it's got a ton of different names. And basically it goes in with acetyl-CoA and it joins with oxaloacetate, which is that 4-carbon molecule. And that's going to form the citric, right? That's the 6 carbon. And some people call it citrate because it's part of the citric acid. cycle. And then that citrate gets turned into a whole bunch of other things, because the cycle starts off with 4, right, which is the oxaloacetate, and then it gets turned back to that to maintain the cycle you go through. So basically, the gist of it's 1 ATP, 3 NADH, and 1 FADH2. And you're going to double this because if you're counting a glucose. All right, so the best way to do this, and the last one here, of course, is oxidative phosphorylation. Gives you these. So you have a total of 12 electron energy carriers, if you will. And these guys are going to be shuttling back and forth in an electron transport chain. The resulting is the NADH, or the NAD and the FADH are going to be recycled back, and they're going to be your carriers again. And we're going to go off to the races. Okay. This thing gets passed down. Anything that you might get in terms of a question that's going to ask or that's going to inhibit one of these stages, you need to think about. For example, one of the things that could occur, I don't think you've seen this, anything that blocks this obviously is gonna create issues. The electron transport chain, for example, certain poisons would stop that. And if I remember right, and I'm doing this from memory, I think it's cyanide, something like that. So that would be an example. But again, any kind of substance, if you ever got a question about that, where is this in the pathway? You could figure out something like that. Okay. Okay, very good. So in summary, basically, the process of aerobic would be these 4 steps. And this chart basically summarizes the total overall net that you're going to get in terms of the ATP as you go through. So knowing this again and studying this and being able to have it, in your knowledge base would be fantastic. Okay. Photosynthesis versus cellular respiration is also very important. Okay. And this is just a couple of notes on that. Basically, you have a proton gradient where you're dealing with, in both cases, and gradients are very important. When you're dealing with an impetus to have something, a discussion of a gradient is very important. In this case, the proton gradient is the electron transport chain, okay? And in respiration, protons are pumped, right, from the mitochondrial matrix into the space. Anaerobic has to do with lactic acid, right, or ethanol, and fermentation. So be on the aware of that and understand basically what happens with your exercise. You're going to have a buildup, right? And you're going to have, obviously, you've heard of lactic acid, and this basically switch over anaerobic, pyruvic, produced from the glycolysis is converted to lactic, and that's what happens when you exercise. All right, so in summary, we talked about energy, enzymes, okay? And again, we get it, that's a lot, there's a lot of information, but the key thing here is to just organize it and to be able to take it and compartmentalize like you would in biology and compartmentalize it all put together, and then little by little, all these pieces will fall into place in terms of what you need to know. Okay, and then cellular respiration, we gave a very overview, a very cursory overview, if you will, in terms of that. Okay. A couple of just more words before I get into one more topic. This is just talking about communication between cells. We talked about the energetics, but now talk about this. Chemotaxis is movement. You have ligands, some people pronounce it ligands. We've always argued about that. Signal transduction is very, very important in terms of this, where you're going to have signaling, activation, and then a production of a response. Obviously, things like this is big in pharmaceuticals and so forth. You have different types of membrane receptors, whether they're the gated or the catalytic or the G protein linked receptors. Homeostasis is your steady state. When you say steady state, if you're ever going to explain it to anyone, you wouldn't be able to do this on the test because they wouldn't want you to spend a long time of explaining it. But if you had your bathtub faucet on and the rate by which water flows out of the faucet and the rate by which the water drains, into the piping, If you get that rate, those rates together, then you have a steady state. That's just one example, but steady state's important. It doesn't mean that the rates are zero. It means that the rates are equal enough in terms of what you're trying to maintain in terms of homeostasis. And of course, homeostasis is very important in blood glucose levels in pancreas. You either have a negative feedback or a positive feedback, depending whether you're going to have inhibiting or a stimulation, obviously, when you have a positive feedback. Communication in plants, they generate electrical signals in response to stimuli. So this is just more of an explanation in terms of understanding the communication that you have. Okay? So hopefully that's good. The last thing I want to talk about, and I had somebody alluded to this last time, is a discussion on qualitative skills that you need to know. Now, like I said, this is a good time to bring this up because it's something that you can consider. One of the things that you can have overall in terms of that you would absolutely benefit from is a full understanding of quantitative skills. It does not mean that you have to be a biostats wizard, because in some of these problems, all right, and even though you're going to get two, you're guaranteed one of them is going to be something where you're going to have to, where they may, you know, ask you some kind of stats. Does it mean that you absolutely have to, you know, generate the formulas? Well, the formulas are given to you in the formula sheet that you have, some basic stuff. But let's have an overview in terms of understanding the ideas behind this, and we'll do that in the remaining time. Okay, you'll see it in different things like water potential, etc. But here's the key. Let's start simply. Okay, let's read and predict and understand the basics of that. Okay. Sometimes you're going to see a bar graph. Okay, they call these a whole bunch of different things, column charts, etc. This is looking at fly populations in northern Maine. Okay, some of the things that you want to do when you have a table and a chart, now this is very basic knowledge, but it's also very useful, is to get your bearings. Look at the title, look at the axes, note the legend. Okay, make sure you know the legend, the key. Make sure you fully understand, well, I say fully, but I mean you have a good handle on what it is that you have here. Okay, now in terms of some of these, in this kind of thing, this being a column graph, all right, basically it's trying to tell you three different populations, okay, three different species within the same month, all right, separated in terms of this. Okay, sometimes you're going to see data that's going to look like this in terms of frequencies, and they're going to have it plotted. Again, you still need to note frequency, note height, note the title, and what the difference is between these. Okay, there are different things that are discussed here. Right? This is called normal parametrics, and this is just basically data that forms a normal curve. A normal curve has many names: Gaussian, bell curve, normal curve, etc. But basically, if you have a large sample size, this is basically what you're going to see. If you took a large sample of ages or whatever the case might be and plotted, in this case, heights, because that's what it's saying, heights, you would get this kind of relationship. Okay? Now, the basics of understanding the scientific method, you have a hypothesis. That's going to be the thing that's a well thought out. You have your independent variable, the thing that you're going to change between the groups. And then you got your dependent, which is dependent on the independent variable. Okay, so in other words, it's the thing that you're going to get. You can consider x comma y an ordered pair. You punch in an x and you get out the y. Okay, you should know what a constant is, a control variable. You should know a control group, right? The one that's got no treatment. You need the control group for comparison's sake. And then when you hear the word statistical significance, which you can also use, it's also basically saying that you have a certainty in your conclusions. Okay, you will always be able to do better if you increase your group size and include more trials. That's a given. Bioexperiments, or the problems that you're going to see, are either going to look at how something changes over time, compare, or test for some type of association. If you were looking at something like this, this is a line graph. Okay, Now, what's important in line graphs, these are time course experiments. So over time, you see some kind of change. Nothing changes. You still look at the title, look at the axes, and denote whatever's occurring in terms of that. Okay? Sometimes you're going to have to go a little bit further in terms of that. Okay? Sometimes you're going to have to go and look at mean values and medians and everything else. You can refer to the basics of mean being the average, median being the middle, and the mode being for the most in terms of that. And this is just an explanation in terms of the kind of things that you'll see. What's important here is understanding the key things. You probably won't be asked to calculate a mean. That's pretty straightforward and easy. And the median and the mode, okay, like we said. And this is just an example. But to understand a standard error, which is a deviation, standard deviation is basically the spread of the data, okay? And easy way to remember standard deviation is basically something like if I took a test, I told you if I gave you a test and I told you the average was an 80, then you should ask me what's the standard deviation, because I could tell you, well, half the class got a 60 and the other class got 100. Well, that's an average of an 80. But there's something wrong with my test if half the group failed and the other half got a perfect score. So something's wrong. The standard deviation's high. But if I have a standard deviation that's lower, then some people got 77, 78s, 83s, 82s, and the average is still 80, but the deviation is a lot smaller, and then I won't feel as bad as if something was wrong. And that's the gist of that. Well, with the standard error is the standard deviation divided by a square root in terms of the sample size. And in this case, you just have to know what the standard or what the sample size is, and you should be okay. Larger deviation means the data are more variable, okay? And with most of these sets, you want to look at a normal distribution and arrive at the comfortable conclusions in terms of that. There's also, and remember there's a t-test and a p-test, and I glossed over that, but basically, if you get a p-value that's equal to or below 0.05, that's considered significant in most of the bio fields. That means that there is some kind of effect that's there. T-test calculations involve comparing each data point to the group's mean in terms of that. Now, you also have things called chi-squares, which is used to figure out observed and expected data. It starts off with a null hypothesis, which is what you're saying the outcome is going to be that you want to compare with. You're always going to fall into that as the default, okay? Now, you want to do this chi-square test to see if your data is actually going to be close to this outcome. If it's either going to be close to the outcome, or you could say it occurred due to some kind of random occurrence, okay? So you basically are trying to say, we use the word fluke here. Is it a fluke or not that you have this? And this is a good example of a test of how you would want to do that. Okay, this one here involves everything, and I understand that we could look at this one here just so that you have a decent example of this. If you had data collected from 100 families, you had 3 children each, 14 of them had 3, 36 had 2, and so forth, then you want to, you would expect offspring to segregate independently, right? And we'll talk more about that next week. In other words, the sex of a given child shouldn't be affected by the sex of the previous children. Now you want to test that in terms of that, okay? So you're saying that sex is segregated independently. That would be your null, right? H naught. And each family is going to have 3 children, so there's only 4 outcomes: 3 daughters, 2 sons, 1 daughter, or 3 sons. Any of these 4, okay? And you don't need to worry about the math, but you can figure out these percentages. What are the probabilities, which you will not have to do on this test. So because there's 100 families, you expect 12%, given these numbers, again, that you don't need to calculate, to have 3 females and so forth, okay? So the way you would do this is the probability of something that occurs times the actual event is what they call an expected value. Anyway, so you have expected data to actually what happened. So O is your observed versus what you would expect. Your expected is what we talked about just here when you got your numbers. Observed is what the data are in terms of what you got. You calculate this quick thing, all right, and then you summate. Okay. You have a 6.24, and then you need to compare it to something called a CV, or a critical value, which you would obtain from a table that's given here, which you would be provided with. And you must know what degrees of freedom is, which basically it says how many independent variables are in the data. So degrees of freedom in this case, -1, because you're comparing 4 different family types. Okay. And then you need a reference p-value, which for most of things 0.05 is used. So then when you have degrees of freedom is 3 and a p-value of 0.05, you find the critical value according to the chart, and then you compare it to your critical value. Okay, or I should say your chi-squared value. This is your chi-squared, right? The Greek letter chi. So if your chi-square is greater than the critical value, you reject. So that's the only thing that you need to know in terms of this. So 6.24, which is your chi value based on everything that you have calculated, compared to 7.82, notice that that is less than the critical value, so you accept the null hypothesis. If you accept the null hypothesis, then what you stated, it's not that you go to the alternate, you just say offspring sex could indeed be segregating independently, and that's basically the only conclusion that you can come across. So with descriptive, in summary, descriptive data, you basically have to put all this together, normal, parametrics. Things like histograms are basically bins and buckets. Counting data is in a bar or a pie chart. Pie graph is nothing more than just a pizza slice, as we call it, where it's just percentages. You can't get trends really that well with a pie chart. Time course experiments is where you're going to get your charts, and experiments that are going to compare groups with data standard deviations is very key in terms of chi-squares and so forth. Okay. Scatter plots mean you just throw the data onto an XY plot and see if you can get any kind of linear or curvilinear thing. Probability is other things that you can look up, but summate, whether it's the probability of this and this, or the probability of this or this. It's a matter of either addition or multiplication. And hypothesis testing already we mentioned with the p-value and the chi-square. I want to thank you very much for everything. I get it that there's a tremendous amount of material. Feel free to email me. I want to thank Shadna and Brian and my helpers today. And I want to wish you the absolute best of luck in your journey through this, as well as everything else. Please stay safe, stay healthy. If there's any questions, you can either email me, or you can go to our website, www.princetonreview.com. Again, best of luck. And if there's any way I can help you, we're here to help you. All right, take care. Have a great day.